Hello everyone, today we will talk about the lipids. What are lipids? Lipids are chemically diverse organic compounds which perform diverse functions. And very common thing about all the lipids are they are organic compound which are hydrophobic, means they are insoluble in water. Hydro means water, phobic means fear, so they repel water. But they are soluble in non-polar solvents such as chloroform. What are the function of lipids? They are a stored form of energy like fats and oils. They are structural elements like phospholipids and sterols, as phospholipids form the bilayer of plasma membrane. They also plays other crucial roles like they are enzyme cofactors, electron carriers, light absorbing pigments, vitamins, hormones and intracellular messengers. Lipids are classified into three groups. First one are simple lipids, second one are compound lipids and third one are derived lipids. Simple lipids are composed of fatty acids and alcohol. Compound lipids have, apart from fatty acid and alcohol, they also have additional groups, and derived lipids are hydrolysis product of simple and compound lipids. So basically derived lipids are product of simple and compound lipids, and they have basic characteristic of lipid, that is they are also insoluble in water. Example of simple lipids are triacylglycerol. Triacylglycerol are composed of glycerol and fatty acid. So glycerol is alcohol here and fatty acid is present. Next example are waxes. Waxes are also simple lipids. They are also made up of fatty acid chain and they have long chain alcohol. So there is a difference between triacylglycerol and waxes is that in triacylglycerol we have glycerol is as alcohol, present as alcohol, and in waxes they have different alcohol which are very long. Glycerol is very small alcohol with only 3 carbon atom. Next one are compound lipids. Example of compound lipids are phospholipids, glycolipids and lipoproteins. So all of them have fatty acid and alcohol. In phospholipid, additional group present is phosphate. In glycolipids, additional group is carbohydrate. In lipoproteins, additional group is protein. Example of derived lipids are fatty acid, steroids, eicosanoids, fat-soluble vitamins like A, D, E and K, and ketone bodies. In this lecture, we will talk about the simple lipids. They are made up of fatty acids and alcohol, and examples are triacylglycerols. They are storage lipids. In our body, fats are stored in the form of triacylglycerols. Like lipids are stored as triacylglycerols. So whenever we require long-term energy, these lipids are used. Next are waxes. Waxes are a storage lipid as well as protective lipids. For example, you must have heard about the bee wax or ear wax. All these waxes are example of simple lipids. Let's first talk about the triacylglycerol. They are also called as triglycerides or neutral fats. They are fatty acid esters of glycerol. Let's understand this term one by one. So this is the glycerol. This is 3 carbon alcohol. These are hydroxyl groups present in glycerol, and this is fatty acid chain. So these are 3 different fatty acid chains. This is one fatty acid chain. So here this is hydrocarbon chain made up of carbon hydrogen, and here this is carboxylic group. So this carboxylic group is polar, and this part hydrocarbon chain is non-polar. Because of presence of this carboxylic acid, these are called as fatty acid. They are carboxylic acid of hydrocarbons, and this is polar and this is non-polar, and because of that fatty acids are amphipathic, means they can half part is can interact with water and this part hate water. Now with glycerol, this all the OH part of this will interact with this carboxyl part of this fatty acid. So one H from here and one OH from here will be released as water, and these two molecules, this glycerol and these three fatty acid, will be interacting with ester bond. This bond is ester bond present here. That's why their triglycerols are called as fatty acid esters of glycerol. So here this R1 is this fatty acid chain, this C double bond comes from here, this carboxylic acid, and this O comes from here. As this OH and this H is released as water, and this process is called dehydration or condensation. We can break triacylglycerol by the process of hydrolysis, means when you provide water and do the lysis, this is called hydrolysis. So by doing hydrolysis of triacylglycerol, we will again get glycerol and 3 fatty acids. Example of this is the example of triacylglycerol. So how we name them? This is at first position we have one type of fatty acid is called palmitoyl. At second position we have different fatty acid, linalool. At third position here we have different fatty acid, that is esteriol, and then we name it glycerol. So this way we can name a triacylglycerol. Now you need to remember that it's not necessary that all the 3 chain will be different or identical. They can be identical, they can all be different, and 2 can be same, 1 will be different. So that way many different types of fatty acid combine with glycerol and form different types of triacylglycerols. Examples of triacylglycerol are fats. Fats are solid at room temperature. So example is butter, and oils. Oil are liquid at room temperature. Examples are corn oil and olive oil. So question is, if fats and oils both are triacylglycerol, why one is solid and other is liquid? So answer is fatty acid. Fatty acid can be of different types. So let's talk about fatty acid. Fatty acids are carboxylic acid, we already discussed in previous slide, and with long hydrocarbon chain. Here this is hydrocarbon chain, this carboxylic acid, and we can label them. We always name from this carbon functional group carboxylic acid. It will be first carbon, then second, and then and so on. So this is 12 number carbon fatty acid. Fatty acid are empipathic because this is polar group and this is non-polar group. Usually they have even number of carbon. Here I have shown the example of 12, and another they can be 12, 14, 18, 16, 20, 24. It is usually, means most common fatty acid are have even number of carbon, but it's not always necessary. Some exception are also there. So fatty acid also exist in odd number carbon with odd number carbon atoms. They are usually unbranched. So most commonly they found unbranched, but branched fatty acid also exist. Now we will see the 2 types of fatty acids. One will be saturated, another will be unsaturated. What is the difference between saturated and unsaturated? Saturated means carbon present in fatty acid, all the carbon are engaged or bonded with hydrogen. All carbon, if you see here, are bonded with this hydrogen. In case of unsaturated fatty acid, there will be a double bond or triple bond present. But in case of unsaturation means all carbon are not saturated with hydrogen. Here double bond is present, so 2 hydrogen are lacking here. So that's why they are called unsaturated. In normally, if we talk about general chemistry, in unsaturation there can be double or triple bond. But here in fatty acid, most common bond is double bond. In triple bonds are rare, very rare. Most commonly double bonds are found in unsaturated fatty acid. And double bond also here are not conjugated, means they don't have alternate double bond. Means unsaturated fatty acid can be monounsaturated and polyunsaturated, means they can have single double bond or can have multiple double bond, but conjugated double bond will not be present there. That is also in very rare condition conjugated double bond will also be present. Now, if double bond is present, they can be in cis form or they can be in trans form. Cis form means both the hydrogen are on the same side, so they are cis form, and if both the hydrogen are present in same side, they produce a kink, and you see this is bended fatty acid chain. In case of trans form, both the hydrogen are balanced in opposite direction, so in trans form there will be no kink. And in nature, most commonly present fatty acid, double bond in fatty acid are in cis form. Trans form bonds are rare. Then you must have heard about the trans fat. Then where does it come from? So trans fatty acids are very rare, and transform bonds are introduced by gut bacteria of ruminant animals like cow, sheep, goat, etc., and they can also be produced industrially through a process called partial hydrogenation. So in nature, most common form of double bond present in fatty acids are cis form, but trans form are also introduced by gut bacteria of ruminant animals and industrially through partial hydrogenation. Now let's talk a detail about saturated and unsaturated fatty acids, how they are actually changing the nature of triglycerides. So in saturated fatty acid, they have a straight chain, so they have more chances to interact with each other through van der Waals forces between these two fatty acid chains. So because of that, they are closely placed, they have a straight chain, they can stack on each other and can tightly pack. So they have a stronger interaction, and because of that they have high melting point, and they are usually solid at room temperature. You can compare it with solid. Since solid have closely packed atoms together, they have very high melting point. Similarly here, fatty acid chain are straight, they are interacting closely, they are stacked on each other, so they are forming a stronger interaction. If you look at the unsaturated fatty acid, we discussed that most of the unsaturated fatty acid have double bond here, and because of double bond, cis double bond, they have kink here, and because of this kink they have loose interaction. Because of this kink they cannot pack tightly, so they are loose packing, they have weaker interaction, and because of that they have lower melting point, and that's why they are liquid, usually liquid at room temperature. So, nature of fatty acid can vary on the basis of whether they have a different chain length. Like if it is suppose we have this fatty acid, if we reduce the chain length, we are reducing the interaction between these two fatty acid chain. So here it was interaction all over this length, now this length has reduced, so interaction is less now, so they are now loosely packed and they have lower melting point. If you increase the length of this fatty acid chain, by increasing the length we have more interaction between this chain and they will have higher melting point. So chain length also affect the nature of fatty acid. And second thing which affect the nature of fatty acid is degree of saturation. Like if we have this many unsaturated double bonded fatty acid, then they will be liquid, and when we are mixing both more number of unsaturated like saturated fatty acid, then they will have more solid character and will have higher melting point. And if we have all these saturated fatty acid, then we will have solid character of the fatty acid, and they will have higher melting point. So characteristic or nature of fatty acid can changing by changing the chain length and by changing the degree of saturation. That's why fats and oil have different characters. So oil have many unsaturated fatty acid, and because of that they are liquid at room temperature, and fats have more of saturated fatty acid. They can stack on each other, they can pack tightly, and that's why they are solid at room temperature. These are few examples of saturated fatty acid. As we have discussed, in fatty acid, most common number of carbon present are of even number. So here all these are even number carbon atom containing fatty acid, like 12, 14, 16, 18, 20. And this is the chemical formula. And this is the systematic name. Like if it's with 12 carbon, we call it decanoic acid. Do means 2, deca means 10, decanoic acid, and this is lauric acid. And you can see, because of saturated is present here, it is saturated, its melting point is very high, like 44.2 degrees C. And next one is 14 carbon fatty acid, that is tetra. Tetra means 4, decanoic acid. Its common name is myristic acid. It also has higher melting point. 16 carbon fatty acid is palmitic acid. 18 carbon is stearic acid. With 20 carbon is arachidonic acid. You see that this is example of saturated fatty acid, means there is no double bond present here. And if you see, when you are increasing the chain length, number of carbon, we are increasing the number of carbon atom from up to lower side. Here we can see that melting point of this fatty acid is increasing, means increasing the length of carbon atom, means fatty acid chain, we are increasing the melting temperature. Next one are unsaturated fatty acid. Examples are, first one is palmitoleic acid. Palmitoleic acid is 16 carbon unsaturated fatty acid. Here delta represent the double bonds and 9 represent the position of double bond. So here only single double bond is present at 9th position. So this is it can be written as 16 1 delta 9. And you can see melting point is very low here because it's unsaturated fatty acid. Next one is, this is 18 carbon fatty acid. Here also double bond is at 9th position, means here chain length has increased and single double bond is present here, and this is oleic acid. Next one is 18 chain carbon fatty acid. It is also 18 carbon, but here 2 double bond are present at 9th and 12th position, and this is called linoleic acid. And here you can see that here at 9th position this double bond is, this is the structure of linoleic acid. At 9th position we have 1 double bond, at 12th position we have double bond. Fourth one is linoleic acid. It also has 18th carbon and 18 carbon, and 3 double bonds are present at 9th, 12th and 15th position. So you can just remember 1 double bond position like 9th number, and you can increase by just adding 3. Like here you add 9 plus 3, 9 and 12 in linoleic acid, then you add 9 plus 3 plus 12, and then 12 plus 3 is 15. So 3 bond will be alternate of 3. In case of arachidonic acid, 4 double bond are present, and it is 20 carbon atom fatty acid chain. So here, since we are increasing the length and these are unsaturated fatty acid, they have lower melting points. So this was all about the triacylglycerols. Now next example of simple lipids are waxes. Waxes are also similar to triacylglycerol. They also have alcohol and fatty acid. Here also long fatty acid chains are present, and here alcohol is also very large. In case of triacylglycerol, only 3 carbon alcohol glycerol was present, but here we have long chain of alcohols are present. Example is bee wax. Bee wax is major component of bee wax is triacyltanoyl palmitate, and this is the ester of palmitic acid with alcohol triacyltanol. If you look here, palmitic acid is 16 carbon long fatty acid chain, and this triacyltanol is very long, like 30 carbon long alcohol. So in waxes, fatty acid as well as alcohol are very long chain. Other example of waxes are some planktons use waxes as storage lipids. Here in beeswax, these waxes are protective lipids. They are not a storage lipid, but in some planktons, waxes are used as storage lipids. In earwax, we have it's also a simple lipid, and it is a protective lipid. It provides protection. So this was all about the triacylglycerols and waxes. So today we finished the simple lipids. In next lecture we will talk about the compound lipids. Thanks for watching. See you in the next video.